First of all, thanks for the SACS conference to organizing this usually very, very exciting meeting. Thanks also for the audience who stayed with us despite of being rather late now. Two housekeeping points. I would like to ask the presenters to keep the time. So this is usually 3 minutes for presentation, 2 minutes reserved for questions so that we stay in time. And I have an ask for the audience as well. Please follow the presentation carefully and if possible, ask really important questions. You have the opportunity, of course, to meet the presenters afterwards if you have further burning questions. So that said, I think we should start with our first presenter. And this is Ian Levine from ModernVivo from Seattle, who is talking about a new way of evaluating information from in vivo experiments.
Thank you so much, and thank you so much for having me. Happy to tell you a little bit about ModernVivo. I'm Ian Levine. I'm the co-founder and CEO of ModernVivo, where our mission is to help enable better drug discovery through AI-optimized in vivo models. So anyone in the room who's done in vivo work in the past knows that the process of designing these models can be a very slow and tedious process, where you're querying databases of literature such as PubMed or Google Scholar to identify what models are relevant for my disease indication or my target of interest, where oftentimes you're either getting thousands of results or thousands of papers or maybe a very few number of papers that are not really relevant. So this process of actually reading these papers can actually take months of time to actually design these models. And this process has really remained unchanged for decades. So what we've developed at ModernVivo is an AI-powered protocol design software where all you have to do as the scientist is upload some kind of information around your experiment of interest, whether this be a target, a therapeutic class, a cell line, a biomarker, can be as broad or as specific as what you want. What we do then is we identify and analyze all published papers to identify what experimental models are relevant for your therapeutic of interest. What we do then is index every single paper that is relevant to that particular model, extract the protocol conditions directly from that paper so that you can plug this information directly into your protocol. We also have methods of analyzing what is the reproducibility of these models, how oftentimes these experimental designs have been cited in the past, have these experimental designs been used to justify clinical trials. Our technology analyzes the full scope of the literature, not just abstracts and footnotes. We have AI-powered patent-pending technology, and we have a novel way of indexing this experimental data that manual human review and general AI tools are not able to do. We've worked with over 30 different pharmaceutical companies, from the smallest biotechs up to some of the largest pharmaceutical companies, and also have contracts with 10 CROs across all disease indications. We have a team that is a mix of not just life science experts like myself, who my expertise is in neuroscience and oncology, but also a world-class team of machine learning engineers, and as well as an incredible advisory board. So finishing up a few seconds early, we look forward to working with any size biotech company, from the smallest all the way up to some of the world's largest. We just closed the financing round, so not necessarily need yet, but I'm sure we will soon. And then, happy to answer any questions. So thank you.
Thank you very much, Ian. We have a question from Esteban.
One question. How do you avoid hallucinations in your AI system?
Yeah, that's a great question. So without going too technical here, we've been around for 3 years, but have only been in market for a year because we took that 2 years to make sure that our technology is not hallucinating. Our software, as it was built specifically for in vivo scientists, was trained specifically on in vivo terminology. And that's what our intellectual property is around, is identifying not just how in vivo specific terminology is published, but how it is published differently across different publications. But to answer your question more directly, we have QA constantly making sure that we're not hallucinating. It's a continuous effort. It's not just it worked well once and we're good going forward. And we found that we're actually above manual human review accuracy.
Our next speaker is Patrick Hamelin, who is with Arivin Therapeutics from Helsinki, Finland. And the company is developing new therapies against bacterial infections, especially resistant antimicrobial infections. So Patrick.
Good afternoon. I'm Patrick. The CBO of Arivin Therapeutics. We develop future-proof therapies to halt resistant infections. So antibiotics are failing in treatment of infections because the bacterial pathogens develop resistance at the faster pace than we are able to develop new effective antibiotics. And some 20 years ago, we encountered the first pan-drug resistant infections, and these then are resistant to all antibiotics in all different classes, and has made bacterial infections far more deadly. And the resistant infections actually kill more than 1 million people annually, and this is really expected to grow rapidly up to 10 million by 2050, which would be the level of all cancers together. For decades, the efforts have been mainly put on developing therapies that are targeting the bacterial pathogens as such, mostly then by antibiotics. However, as said, the resistance development makes this nearly impossible to find new innovations. At Arivin, we take a different turn by targeting the bacterial virulence factors, which the bacteria produce and which make you ill. And we believe this is a more effective way to treat these infections. We developed this novel antivirulence therapy that has a dual mechanism of action, binding the bacterial toxins and breaking down the protective biofilms. And this lowers the ability of the bacteria to cause the tissue damage and also to establish and spread the infections. There are two great benefits of these therapies over antibiotics. Firstly, they are effective on the pan-drug resistant pathogens. And secondly, also, as we target something which is outside the bacteria, these pathogens do not develop rapid resistance against this therapy. So that's why we call them future-proof. So in our lead program, we focus on applying this on respiratory infections. So cystic fibrosis and NCFB have significant patient populations. And despite there are available treatments and they are expensive, some 50 to 70% of these patients suffer from long-term chronic infections and exacerbations. Our therapies are addressing this unmet need by significantly reducing the inflammation and the chronic colonization of the bacteria, which we can show in our in vivo models. We have strong IPR. We have 4 patent families and already 3 granted patents in the US. The lead program we have is in the preclinical IND enabling stage, and we have recently completed a seed financing round, enabling us to reach first in man by early 2027. We are looking at the business opportunity, which we believe would have more than $1 billion of peak revenue potential, and certainly then are looking for Series A participation by investors at late 2026. So please join us in our mission to take this therapy into life-saving use in the future.
What is this FastP screen platform? Could you shortly introduce it to us?
Sure. So that's kind of a third asset we're thinking about in developing this into a platform, and that would be basically to identify new lead molecules for either in cooperation and of course serves our own pipeline development as well.
Thank you very much, Patrick. So, an applause, please. And our next speaker is Stephanie Holstein-Rönsbo, who is the CEO of Loma Therapeutics. They are developing a new wave of HPV vaccines, is my understanding. HPV has been known and has been around for a long time, known for causing cancer and other conditions. So, Stephanie, please.
Thank you so much. So my name is Stephanie and I'm the co-founder of Loma Therapeutics. We are developing an immunotherapy against HPV-positive cancers with best-in-class potential. So we are targeting HPV cancer because it's unfortunately not a finished chapter, despite an excellent preventive vaccine on the market. In fact, for oropharyngeal cancer cases, which is the indication that Loma is focusing on, the numbers, as you can see, are actually increasing by 60% over the coming 30 years. When a patient is treated, the person might have some HPV remnants in them, and a quarter of the treated patients will experience a relapse of their cancer. And in that case, they have a really poor prognosis of less than a year and really limited treatment options. So obviously there's a need for new therapeutics in this space, and this is where Loma comes into the picture. We are developing an HPV-specific immunotherapy, and the unique design you can see here is incorporating a lot of HPV material. And the vast amount of epitopes here to activate the immune response of our patients will induce a really broad response in the patients. We will secure an effect in many more individual patients, and this type of technology has an excellent safety profile. We have treated mice that have been given HPV-16 cancer cells. And what you see here is that we have a synergistic effect with chemotherapy, both in terms of the tumor size and the survival of the mice. What's even more interesting is that when we benchmark that to competitive solutions that are targeting exclusively E6 and E7, we can see that the added amount of HPV material, so in this case E1 and E2, is really enhancing the tumor control and the survival of the mice. And this is what we want to bring to our patients. Our phase 1 is designed and based on a biomarker, and the biomarker will reveal with almost 100% certainty whether or not this patient will face a relapse of their cancer. So the use of this biomarker is really powering our phase 1 study effectively. Our phase 2 is currently planned to be pivotal so that we can apply for an accelerated approval. And to get us further, we are currently fundraising for our Series A to get us through the phase 1. We envision an exit after the phase 2. It doesn't stop with oropharyngeal cancer. We envision a portfolio potential. We can treat any other HPV-16 positive cancer, as well as develop new drugs also targeting other highly aggressive HPV types. This is the wonderful team behind Loma, the day-to-day operational team, our wonderful board of directors, and a close range of key opinion leaders within HPV and viral vector technology. I hope that you will join us in our mission. Thank you.
Thank you very much. Excellent presentation. May I start with one question here?
Yes.
So you are putting really quite a bit of the viral genome into your vaccine. Can you control a potential recombination within the patient and ensure that no wild-type viruses are actually amplified?
Yes, so we have, of course, included some specific mutations that render this a safe treatment for the patient. So this will be presented to the antigen-preventing cells and activate the T cells, but there's no risk of causing HPV disease in our patients.
And our next speaker is Philip Busenhardt, who is coming from Adularia. And Adularia is telling us about their experience, which they made in microbiome, converted into a cancer therapy. So a very exciting topic.
Thank you very much, Tamas. Yes, we are Adularia. We're developing cancer immunotherapies. And as you know, cancer immunotherapies have been a huge success in recent years. However, 85% of patients do not respond to cancer immunotherapies. And out of the responders, less than 20% are actually long-term remission responders. So there's still a huge need to develop new approaches to improve cancer immunotherapies. Our approach started with analysis of the microbiome, in which we found that certain bacterial species of the microbiome are underrepresented in cancer patients when compared to healthy individuals. So we then took these bacterial species and orally applied them to mice or to mouse models of cancer to see if there's a causative effect with these bacteria. And what we found was that the mice that have received these bacteria had a very strongly reduced tumor growth. And when we then analyzed the blood or the serum of these mice, we identified a specific bacterial metabolite that was produced by these bacteria. We then applied only the metabolite alone and found the very same effect with this bacterial metabolite that we have seen before with the bacteria. As the next step, we then set out to identify the target of this bacterial metabolite, which we identified to be the GPR84 receptor. And we tested this by applying the metabolite together with an antagonist for GPR84. And by doing that, we could completely abolish the effect of this bacterial metabolite. So what does GPR84 do? It is expressed on macrophages. And once it's activated, or once the macrophage get activated through GPR84 activation, they start to produce cytokines, like in this example IL-12, which then recruits T cells and activates them, and then they can infiltrate into the tumor mass and kill the tumor cells. So what we're doing now is we developed small molecules to target this GPR84 receptor. And we have developed multiple compound series now with very high potencies, and have now a very promising lead candidate that has a very high potency for GPR84 activation. It has a very good half-life that supports once daily dosing. It has oral bioavailability, so we can actually apply this compound orally. It has a simple synthesis pathway for a simple scale-up of the production. Currently, we have our animal studies ongoing with these lead compounds. We just this week received some very promising intermediate results. And we have filed the patent for this lead compound series, and will soon file patents for 2 backup compound series. So currently, we're still optimizing these leads, performed in vivo studies, and by mid next year, we want to have selected the final lead, which we go into manufacturing scale-up and dose range finding studies with. For these activities, we're now seeking for a seed funding round. Then by end of 2027, we want to reach the IND, and then in 2028 start our clinical trials.
So, Philip,
Yeah.
thank you very much.
Thank
you for time for the presentation. Any questions from the audience, please? This is a very intriguing topic, so I'm sure there will be interesting ideas around it.
Great presentation. Why generate the small molecule when you could just administer the bacteria that you identified?
Instead of the bacteria, you mean? Well, the bacteria is a much more complicated system. The initial idea was actually to develop the bacteria like as a LBP. We realized very soon that there's very limited interest among pharma and investors for these LBP approaches, because the systems are just too complicated and you have too many variables that you cannot really control very well. And having identified the target of these bacteria, we have a much more targeted approach. It's much simpler to develop.
Thank you very much. Excellent response. Our next speaker is Valentina Garanzi from Diamante. And this is a company about unique plant-based nanomaterials which can restore immune tolerance and especially provide a treatment for diseases like rheumatoid arthritis. So Valentina, please.
Thank you. Good morning, everyone. My name is Valentina Garonzi and I'm the CEO of Diamante, a female-led biotechnology company that is rewriting the way to treat autoimmune diseases. So we have developed a platform technology to produce drugs that is able to re-educate the human immune system, and we are producing these drugs into the plants. So our lead candidate now targets rheumatoid arthritis, and we are planning to do phase 1 next year. And to date, we have already secured 4.5 million euros through grants and international investors. And we are protecting our technology with 6 patents, of which one that is related to rheumatoid arthritis is already allowed into the United States. So now we are looking to raise 10 million, and we have already secured 7.5 million through public funds, and we are looking for 3 million from a private investor. But let's take a step back. Why autoimmune diseases? Because autoimmune diseases are a complex disease. There is no cure. They are treated by a general immunosuppression approach, and this causes a lot of severe side effects. So there is still a huge unmet need for more secure treatment. So this is what Diamante is offering. Our drug is based on the composition of a plant virus plus an immunodominant peptide, and the composition of both are able to induce tolerance in the human immune system by regulation the T cells and with a balance of the cytokine profile. So this mechanism of action is patented and is expandable in other autoimmune diseases. We have tested this approach in 5 preclinical trials for rheumatoid arthritis, where we obtained the complete remission of the disease, no side effects, and superior clinical score than the golden standard. So this is our roadmap, ambitious but clear. We are approaching toxicology studies. We have already a GMP-like facility that we are going to certify next year, and we are planning to do a phase 1 in Barcelona next year, and then phase 2 in early treated patients. So we expect a license agreement in 2028. This is our team. I'm responsible for the business development. Linda Vesani, biotechnologist, she's a high-level expert in this area. And then we have a team also in the operation. And we are also supported by an advisory board on well-established expert in different areas, rheumatologists. We have also Guido Rasi, ex-EMA, that is supporting us in the regulatory aspects, and hematologists and expert in the drug development. So we believe that the re-education of the human immune system is becoming a main industry pillar, and we want to pioneer it. So we invite you in joining us. Thank you.
Thank you very much, Valentina. Questions? I was actually very much intrigued by this notion that you have an immunomodulatory peptide. This has been for a long time a holy grail in the autoimmune field. So can you reveal a little bit more about what this peptide is and how did you get there?
Yeah, it's an immunodominant peptide for rheumatoid arthritis, and we know that peptides alone are not effective to trigger tolerance induction. In fact, we tested also alone and we didn't have any result, but the combination with the plant virus works. So the advantages of using plants is only because it's possible to use this plant virus plus the peptide. And we saw that only the combination really is able to induce tolerance.
So let's move on. Our next speaker is Elisa Roxat from Chromos Labs, and she is coming to us from Australia and presenting a wonderful new technology which can be helpful for monitoring CNS and other diseases.
Thank you very much, Thomas. Hi, everyone. Today, I'm going to share with you how our quantum technology can create opportunities to understand neurological disorders better and how drugs affect on human brain function in preclinical screening. So you see, I'm sure that we've heard a lot about how neurological disorders at the moment causing lots of burden in societies. The problem is that for most of the neurological disorders, there is no ultimate cure, and patients are always taking pills to controlling their symptoms. 95% of clinical trials for neuropharmaceuticals are failing, and this is mostly because there is no solid preclinical model or screening that can capture the complexity of neuronal network for human brain. Even a mild, modest way to improve success rate can save billions of dollars and lots of time. But how can we do this? We have a quantum technology based on diamonds, and these diamonds are only a few millimeters in dimension and can host neuronal cell cultures. Once they capture the cell cultures, the quantum transporters in the diamond can record the tiniest electrical activity of neuron cells and the whole neuronal network. Our technology is label-free and non-invasive, and we want to take the data that it provides us for us to study the whole neuronal network and how it's functioning. Our platform is going to be the first ever to record and show how the disease fingerprint shifts as drug starts to affecting on that culture and neuronal cells. And of course that we have competitors, but we're standing ahead of them by being easy to use, with high resolution and 1,000 times greater to scale. We already measured single cell, and we're looking to imaging the whole neuronal network to distinguish between diseased neuronal culture and a healthy one. And then from there, we're gonna see the effect of drugs on the culture itself. We have an amazing team, top quantum scientists and diamond sensors scientists with myself joining the team. We are looking here for any collaborators who are keen to help us achieving our milestones, and we're partners who would like to help us doing our first raise. Thank you.
Thank you very much, Eliza. So, question from the audience. Personally, I was very impressed when I read your technology. Just to clarify it, my understanding is that what you can do today is in vitro, probably complex cell cultures, but still remaining in vitro. Could you describe a little bit in more detail how that works?
Yes, absolutely. So the good thing is about this technology is that it's not invasive for the patients as well. So you take the iPSC of human neuron cells and you put your culture on a diamond chips, which could be customized for any kind of use, 2 mil or 4 mil, in a well plate, again, as the customer asks for, and you put your neuronal cell cultures on the diamond, and then just a confocal microscopy simple setup can help you image the cell and also the action potentials of the neuronal cell culture.
And since you mentioned sertraline, did you have a chance to profile already drugs in your system and show the efficacy of certain inhibitors or modulators in your system?
That's a great question. It's one of our very close milestones that we are about to achieve.
Okay, thank you very much.
Thank you.
So we continue now. Our next speaker is Xianona Ying, who is co-founder, chair, and CEO of ClearideBio, and she's talking about a bispecific ADC for tumor therapy.
So, hello everyone. I'm glad to introduce you a new co, which hasn't been founded yet, but is planned to be founded in Basel this year. It is a ClearideBio Therapeutics. The mission is simple. We want to drive clarity in chemistry guided by tumor biology, aiming to develop next-generation antibody drug conjugate. So, the ADC is proven to be the most fast-growing drug class in oncology in the past years. It's projected the revenue in this class will be triple in the next decade. However, the current approved ADCs still suffer from safety liabilities and limited patient benefit. So, with Amedioo Therapeutics team, we are aiming to build this new co to drive the next generation ADC to address the current limitations. I have been working in this field over 15 years, going through the proven product since Genmab Tivdak, and also we have been contributed to the Basel-based Amedioo Therapeutics through its acquisition. So over the years, I have worked across 5 different ADC platforms through the early stage to clinic. And my ex-colleague Pava Krom, he's an oncologist, translational scientist, and also a biostatistician. So he has also been working extensively in the field, led about 10 ADC trials globally. And we have also our CFO join us, Daniel. He has 20 plus years of finance and operation global experience. So we have also a group of sound scientific team. Dr. Liang was the co-founder of UK-based Kymab. She has also co-developed the Kymouse platform and had over 100 patents in the antibody field. Lynette is the ex-GSK process chemist expert with 25 years in this field and also supported in the NBE's linker payload development and manufacturing. Sonali is actually based in US. She is ex-Cytomics, which is a very leading masked ADC company, and she has advanced multiple masked ADC programs. So we are very happy to have her as well. So ClearideBio want to try a slightly different business model than what we previously worked as a platform-based innovation-driven ADC companies. So we actually intentionally want to have an asset-centric, platform-adaptive approach by in-licensing early-stage differentiated ADC assets or building blocks. I mean, linker payload, antibody, and so on, through global strategic partnerships. By this multi-platform flexibility, we can de-risk the platform-related risk and then rise the total probability of success of our pipeline.
Would you mind, as a closing step, since we are talking about your first project, to reveal how you have selected your current lead candidate briefly?
Yes, so our lead candidate is a bispecific safer payload ADC, about 15 months to clinic. The lead indication is a squamous lung and prostate cancer. So the target-wise is a worldwide second-in-class. However, the payload has a much safer profile, which we believe would have a clinical benefit. The second program is currently still in the R&D stage, is a masked dual-payload ADC. And so this is basically the pipeline. And basically is based on the three different design polar. One is dual payload, and the other is safer payloads, and then also bispecific or masked antibodies. So yes.
So our next speaker is Michael Schoshan. Who talks about metal-selective peptide therapeutics, and as a first application, Wilson disease.
Great, thank you very much, everyone. Thank you for the panel. I'm very excited to present to you today metaLead Therapeutics, where we aim to pioneer a new era of metal-centered medicine. So metals are pivot to human health. They are extremely significant in various different types of aspects. And of course, the most intuitive one is different types of toxic metals that we should not have in our body, whether intrinsically toxic or essential but misregulated. There's also other categories of metals that are essential, but they might go under deficiency, and this deficiency need to be corrected in order to get normal biology and normal health. And the third category is, of course, something that is growing interest in the last few decades or so, with the growing use of metallic radioisotopes in radioligand therapy and beyond. So generally speaking, metallic therapeutic metals that one should deliver specifically to the target. Now, the main challenge in all of those different categories is our ability to transport and to deliver metals from point A to point B, let it be inside the body or getting it out of the body. That's really the most problematic and the most significant obstacle in this regard. And this is exactly where metaLead comes into play. Our core expertise are with our capabilities of designing specific family of peptides that are capable of binding exclusively to metal ions. The peptides are extremely small, extremely stable, could be administered orally. They could be standalone drugs for various indications, but they could also be a compartment within bigger entities and bigger drug compartments. Now, in terms of metaLead specifically, we've been so far raising 3.6 million Swiss francs from various institutions here in Switzerland and beyond, including Base Lounge and Bioinnovation Institute. And we are now currently raising our seed round of 10 million that will enable us to go all the way to IND submission in our primary indication, including platform expansion. So what kind of peptides are we talking about? Our peptides are indeed having the capability of tailoring metal selectivity and metal specificity, and they are capable of binding to whichever metal that we would like to bind in a tight and effective manner. They are benign, they are orally available, and they are stable and can be functionalized. Now, most importantly, they could be used for all of the different aspects that I've been mentioning, not only shuttling metals and excreting metals out of the body, such as in the case of Wilson disease, but also we are able to deliver metals under deficiency or deliver therapeutic metals into target tissues. And as mentioned, we've been demonstrating already a success proof of concept in vivo validation in Wilson disease, but also in lead poisoning in the past. And we are now moving forward towards the additional indications, including iron overload, including radioligand therapy, and beyond. And this is the team behind metaLead. We are a group of motivated medicinal inorganic chemists, as well as veterans in drug development and beyond, supported by key opinion leaders in the disease areas that we are working in. And we are very much excited to push this program forward. Thank you.
Thank you very much, Nika. There's a question in the audience.
Can you say a little bit more about the application in radioligand therapies?
Of course, yes. So radioligand therapy requires the delivery of a metallic isotope, not always, but most of the times it's metallic radioisotopes into the specific tumor tissue. And the metal isotope must not be leached out and must stay stable before the decay and during the decay. And this is something that currently the standards of care or the chelators that are being used, such as DOTA, DOTAM, and beyond, are incapable of addressing all of the challenges that require such a modality. And metaLead peptides will be able to address the limitations of the current chelators that are out there.
The next company is Mighto Therapeutics, which is looking into mitochondrial diseases. And speaker is Hermann Kaiser, CEO of Mighto.
Yeah, welcome everyone. My name is Hermann. I'm the CEO of Mighto Therapeutics. We're developing small molecule genetic medicines for mitochondrial disorders. So mitochondria are a disease driver behind many disorders, from the rare side, genetic rare diseases like mitochondrial myopathy, to aging-related neurodegenerative diseases like Parkinson's disease. And one of the hallmarks of mitochondrial dysfunction is reduced ATP synthesis that comes about by oxalase decline that's also mediated by mitochondrial DNA that you see in the mitochondrion here. So we're using a very powerful new mechanism of action that works completely different from previous approaches, where we're using a metabolic or proteostatic pathway to selectively boost the biogenesis of functional mitochondria, and at the same time we're discarding and degrading dysfunctional mitochondria. That leads to a general functionality increase in the mitochondrial pool and an overall increase in the fitness. And importantly, this also works on the DNA level, so we see that we can eradicate mutant mitochondria from the cells. So these approach we want to take in two classes of disorders: primary mitochondrial disorders arising by a genetic defect like mitochondrial myopathy, MELAS, MERV, KSS. Global prevalence is 1 in 5,000, no approved treatments whatsoever. And on the other hand, the aging-related neurodegenerative diseases with Parkinson's disease as our lead indication, high global prevalence and symptomatic care only. So in these indications, there's a high unmet medical need, and I've explained earlier kind of what the expansion potential behind therapies are. So we have two programs in our pipeline: MGT200, which is targeting mitochondrial myopathy. We have a clinic-ready asset here that we want to take forward. It's MHRA approved, MHRA reviewed, and the IP comes from University College London. And we have a Parkinson's program, MGT100, with a genetically validated target and IP from University of Massachusetts. On the business side, we're looking here at billion-dollar peak sales opportunities for the orphan drug program, 2.5 billion in the expansion scenario, and also a 4 billion scenario for Parkinson's disease. With that, I'd like to close. So in summary, Mighto Therapeutics is a new biotech company focusing on disease-modifying therapeutics for mitochondrial myopathy and Parkinson's disease. We have a small molecule pipeline, IP from UCL and UMass, clinic-ready asset, and we're collecting a seed round of 13 million to deliver phase 1b efficacy data, lead chemistry for the second program, and build the team. And we have a pharma co-lead investor and followers committed for this seed round. Happy to take your questions.
Thank you very much. So personally, I'm very much impressed by mitochondrial therapies because this is really the energy center of the cell. Have you had a chance to prove that you really have also an impact on basics like NAD production or oxidative phosphorylation as a result of your therapy?
Yes, we did. So we have actually, the work is based on 2 publications, Nature publications from 2021, 2022. Can give you the references, and you can clearly see that we are not only increasing oxalose protein levels, but also respiration rate in these cells, and we also see that in animal models.
So we need to move on. Our next speaker is Nick Siro, who is co-founder and CEO of Serenatis Bio, and they are moving on with a therapy for obsessive compulsive disorder, which is quite a challenge.
Great, thanks very much. So I'm Nick. I'm the co-founder and CEO of Serenatis Bio. We are working on obsessive compulsive disorder. And just last week, we launched our Series A round of $25 million to take 2 of our drugs all the way to phase 1b, to end of phase 1b, and our third one through further discovery and preclinical. So I don't know how much anybody here knows about OCD, but it's a very common disorder. Affects around 3% of the population. 50% of them will be affected severely. And it's not about lining up your pencils, and it's not about color-coding your spreadsheets. It's devastating. I've been facilitating an OCD support group where I live in Cambridge for the past 10 years. This was my best friend called Jim. He had a form of OCD called false memory OCD, where his brain would make up false memories of horrific events that he might have seen on the news, and then he would obsess about whether he'd committed a murder or not. He knew they were false, but they were so strong that he wasn't sure, and that led to taking his own life. Very common, comes up a lot in my support group. Now, there's been no new drug for OCD in the past 30 years. In a previous presentation, they mentioned sertraline. That is the classic OCD drug. For depression, you're given 50 milligrams a day. For OCD, the standard is 200 milligrams a day, sometimes 400, and in extreme situations, 600 milligrams. And you have to wait 3 months to see if they work. Half the time, they don't work. And occasionally, they'll put an antipsychotic on top. Otherwise, you go for very specialist cognitive behavioral therapy. Now, the good thing about OCD, it has the lowest placebo response of all the psychiatric indications. So studies are actually relatively straightforward to do, unlike in depression, okay? And everyone agrees on the endpoints, okay? So we are working on 3 different drugs. Why 3? Because in psychiatry, you never get a drug that works for everybody. The first one comes out of work we've done at the University of Cambridge on the glutamate-GABA imbalance that drives OCD symptoms. We tested a number of mGlu2 modulators in 3 animal models, mouse, rat, and marmoset, and we selected one that's very potent that is now ready for GLP tox phase 1 and phase 1b. Our second drug is an AMPA modulator, which is based on a parent drug developed in France in the 80s. Approved in France, shown to work on obsessive compulsive symptoms, never widely commercialized. We have improved it, increased its half-life tenfold, and got rid of the mu opioid agonism that can lead to abuse. Okay, so it's based on clinical evidence. And this one is also ready for GLP tox and phase 1 and 1b. And our third drug comes out of work I've done with the University of Chicago. I set up a foundation 10 years ago called Orchard that has funded the University of Chicago to test an old drug called tolcapone, a COMT inhibitor. And this has been shown to work in OCD, but tolcapone has a black box warning. So we've done a drug discovery program to synthesize completely new COMT inhibitors that have good membrane permeability and don't get kicked out of the brain by efflux pumps. And that is now ready for further preclinical work. So we use precision psychiatry to EEG biomarkers linked to glutamate GABA ratios that should help us, in theory, predict who responds to the first 2 drugs. And the third one has a very simple saliva test where you test for a certain gene that codes for COMT and that predicts how people respond to that drug. This is our raise. This is our project timeline. And this is the team. So very quickly, myself, I've spent 20 years in drug development. 2 of my boys were diagnosed with a rare disease called alkaptonuria 25 years ago. So I co-founded and set up a consortium, raised 30 million euros, developed a drug through phase 2, phase 3, got EMA approval 5 years ago, and in a separate initiative, got FDA approval this year, and the boys on the drug, it works a treat. So now using those skills, and my co-founder David Kavala is a neuroscientist and a chemist who can do everything from designing new drugs to designing clinical studies. And there you have the rest of the team. These are existing investors. We set up a year ago. We closed our pre-seed round 5 months ago. These are our VCs and a family office and a neuroscience angel. So if anybody's interested in our Series A, please come and speak to me. Thank you very much.
So, thank you very much, Nick. It was really wonderful to hear your story and your energy. In the interest of time, I would like to ask your audience to talk to you directly.
Sure, no problem. Thank you.
If that's okay. And our next speaker is Lasse Reimer from SynuCa Therapeutics. And this is about synucleinopathies, diseases which include like Parkinson's or Lewy body dementia.
Hi everyone, my name is Lasse Reimer. I'm CEO at SynuCa Therapeutics, and we are pioneering the first therapy to slow Parkinson's at its roots. Parkinson's disease is part of an umbrella of disorders called the synucleinopathies. They consist of Parkinson's disease, dementia with Lewy bodies, and multiple system atrophy, which is an orphan disease and our lead indication. They are all age-related neurodegenerative disorders, and they are all pathologically hallmarked by the accumulation and aggregation and spreading of the brain protein called alpha-synuclein. What we have demonstrated is that when you have these pathological forms of alpha-synuclein, they can interact with a specific calcium pump called SynuCa and overactivate it. SynuCa itself is situated in the endoplasmic reticulum membrane, and here it pumps calcium from the cytosol into the ER. But once you have this interaction with aggregated alpha-synuclein, you get an overactivation, meaning that you get more calcium pumped into the ER and also an overflow into the mitochondria. When you have excessive calcium in these organelles, you have a higher ATP production. You get excessive amounts of reactive oxygen species. You get an impairment of autophagy, and eventually all this leads to cell death. So our therapeutic intervention strategy is a small molecule that can tune down the activity of SynuCa down to normal levels. So we have developed SYN4569, which is our lead candidate, and we have administered this compound to 3 different preclinical animal models, 2 in Parkinson's disease and 1 in MSA. We have great efficacy across all models, and today I'll just show you the one in MSA in the sake of time. Here we are administering our small molecule through drinking water, and then we are assessing motor impairment and brain pathology. When we administer this compound, we can delay motor onset, both looking at a clasping score and rotor rod performance, but most surprisingly, that is really nice, is that we can reduce the amount of brain aggregates by more than 90%, which in this model is completely unparalleled. We are right now in a lead optimization stage. We are aiming to nominate a preclinical candidate by next year and a final developmental candidate by 2027. So we are looking to raise seed financing to get us there and get us to the next inflection point. And if any of you are interested in this biology or wish to know more, please reach out to me. Thank you very much.
Thank you very much, Lasse. Any questions from the audience?
Thank you. I'm wondering, how do you test your drugs in preclinical screening?
Yeah, that is a bit tricky, because we have SynuCa as we are interacting with, but we can see that we actually only need extremely low inhibition of the pump to get efficacy, around 2 to 4%. So that is the target validation is one of the things that we have to work a bit more on. We can definitely, of course, see if the compound is getting to the right place, is getting into the CNS. But the target validation is something that we are working hard to find a solution to find that. Yeah.
Thank you very much. Our next company is BIOCELL, and our next speaker is Laura Mann-Kavehasi. So this is about chemoprevention in cancer.
BIOCELL was founded based on long-term research looking for solutions for women at higher risk of breast and ovarian cancer, like myself. We have identified 2 preclinical drug candidates with demonstrated inhibitory effect and high chemoprevention potential, OBX and OPX. There is an urgent need for breast cancer and ovarian cancer risk reduction. Over 50% of demographic are currently at higher risk, more than initially expected. One of the factors being high breast density, and relapses can occur up to 32 years after the end of treatment in close to 50% of the cases. No evidence that ovarian cancer screening saves lives, and no current serious therapy due to chemo resistance. OBX highlights was validated in vitro, estrogen receptor positive, triple negative, and ovarian cancer. Inhibitory effect in vivo, as we can see here. Tumor volume of the treated versus the control group, it's significantly different. Additionally, we have in vitro efficacy compared to tamoxifen, safety record in human use, 16 years of patent life, guaranteed by granted patents in demographics of over 500 million, and it's suitable for long-term prevention. It can be a chemoprevention monotherapy for breast and ovarian cancer. Today, primary chemoprevention for breast cancer, it is recommended by US Preventive Task Force. Secondary chemoprevention ensures proper long-term therapy for up to 30 years to avoid relapse. Ovarian cancer prevention, high need, because no evidence has that screening for ovarian cancer saves lives, and no agent-proven interventional trials to be effective. Expectancy, nothing is done. By 2050, 8 million women can be lost. The drug market sizes for breast, ovarian, and breast cancer prevention is expecting to double within the next decade. Competition, we have the standard of care at the moment, like tamoxifen, raloxifene, and aromatase inhibitors. We know the results of estrogen depletion therapy, poor treatment adherence, side effect, rapidly acquired resistance. No programs currently are evaluated head-to-head with tamoxifen. OPX, prostate cancer chemoprevention and fertility improvement and monotherapy. There is an unmet need for novel agents for prostate cancer chemoprevention. We know that it's actually a lifestyle and metabolic disease. Up to 40% of men will experience relapse after prostate cancer therapy. Low sperm quality and fertility are recognized as early markers for prostate cancer risk. Comparison in vitro with finasteride. And the case study demonstrates that low sperm quality recognized in early studies as prostate cancer risk can be redressed. These are the results of the significant sperm improvement case study. We have the below normal, all the stand parameters here, and we have after treatment very significant increases. This can be an indication for the growing infertility market that's estimated to reach over 5 billion by 2030. The competition at the moment, we're having finasteride and dutasteride with the well-known results and side effects. In particular, finasteride is driving high tumor grade mutation. Our goal is having strategic out-licensing partners in the US, EU, and Japan. We have a quicker route to markets due to FDA regulations for botanical drugs. We have a convenient oral formulation, safety records, and standardized research samples.
So we are moving back to neurodegeneration, and our next speaker is Mighto Therapeutics, who is co-founder and CEO of DCRM Sciences.
Hello everyone, so my name is Mighto Therapeutics, and this is our mechanism of action. So it's AAP, so delta secretase. it's a compound, so we are an inhibitor compound. La Roche also has an inhibitor compound, and one thing to know about AEP is that it cleaves APP and tau proteins, but also alpha-synuclein. So for now, we are mostly doing studies on Alzheimer's, but we have a perspective for also Parkinson's disease. So as you can see, some data on it. So as I said, La Roche has a compound 18, which is also an AEP inhibitor, but theirs is a covalent inhibitor, as ours is a non-covalent inhibitor. And one thing to know is that in full knockout model of AEP, you have, of course, high kidney failures. So that is a problem regarding covalent inhibitors. So as I said, so in Alzheimer's, usually it's 5 big topics that makes you have Alzheimer's. And usually drugs only target 1 out of the 5, but we target 5. So we target the amyloid beta, but also the tau protein, the neuroinflammation, the brain volume reductions, and overall the neuroplasticity and synaptic protein expressions. So here are some insights on the compound. So the compound that we are developing is compound 11A alpha. So those are precursor compound, and those are the let's say old generation that we will soon replace in a way, because the half-life was too low and efflux ratio too high, and this will be finished and better optimized with the compound 11A alpha. We hope to have around 8 to 12 hours of half-life. Here are some data in relation to what I've spoken about. So whether it be for the amyloid beta plaques or the insoluble and soluble tau, as you can see, great reductions. Also, I forgot to mention, but this is an oral pill. And here are some data regarding the NOR test, Morris Water maze, and also regarding the neuroinflammatory markers right there. We can also see in terms of, as I said, the brain volume reduction. As you know, with most amyloid beta focused drugs, you have brain swelling and brain atrophy that can occur. With 11A, do not have any of that, and there is even a prevention of that, which is great. Regarding also the synaptic protein expression, so also really safe at the administered doses. So upon 3 months of chronic use of 11A, no serious toxicity sign has occurred. So it's a really safe drug overall. Here is the competition landscape. You may know, but usually, for example, for Eli Lilly, donanemab makes around 800 million annually last year. If you have a drug with the same efficacy but lower toxicity, you could easily target $8 to $12 billion annual net sales, which we think 11A alpha could definitely trigger. And we believe that 11A alpha is and will be the best Alzheimer's drug candidate on the market. Here is my email if you want to reach out. What we are seeking is partnership opportunities, whether it be licensing or option to license opportunities. Thanks a lot.
Thank you very much, Matteo, also for keeping the time. So I'm sure there are questions from the audience, because Alzheimer's is something which is a rising condition.
So does your drug prevent plaque buildup, or does it clear plaque buildup? From your mouse models, it seems to be that it prevents buildup.
Yeah, so it kind of does both, because we act also on, because there are models that I haven't shown, where Alzheimer's is already instilled and the plaque are reduced by our drug.
If there are no further questions, then thank you very much, Matteo. Our next speaker is Eric Niemela, who is founder and CSO of FINNCURE. And FINNCURE has a repertoire of synthetic carrier particles, which can be used for all sorts of delivery technologies.
Hello, and thank you for having us today. So we are a Finnish biotech company, and we are developing the next generation carriers. And why should you care about that? Well, as you know, many drugs and therapies faces many challenges. Many drugs are very big and complicated, and that leads to solubility issues and permeability issues, or genes that are naked that will be degraded in the body. And all these kind of problems will give rise to low targeting, adverse effect, and all kinds of toxicity. and also stability issues with some RNA vaccines, for example. So we have the solution for this. So we are making a synthetic carrier. So we are actually harnessing the targeting potential of pathogens and making that to a synthetic carrier. And then you can load drugs, API, or genes inside the particle or on top of the particle. And that will target to the tissue of your interest. And then it will be endocytosed and the drug will be then released to the tissue. So it can be more like a vaccine if you wish, more like a drug carrier if you wish, or you can actually use it for blocking infections by having a, let's say, a COVID-like particle sitting on your receptor and then hindering physically the virus from entering. And we have now actually 5 US patents issued and several pendings. So we are a platform. And here you can see that the market is growing. It's going to be insanely big in a few years, like $600 billion US. And here are some data from our in vitro studies, and this was actually published last year, and we came to the front cover of Nanoletters. So the particles are well tolerated in human cells, and you have high uptake, and our microfluidic chip is superior in producing the particles and functionalizing the particles. So that's why it's suitable for this kind of different applications. And last week I came back from Charles River, UK. So we've done several animal studies there, and now this repeated dose study. So we are super happy that you can administer these particles several times to mice, transgenic mice, and no problems. And this picture down here, you can see the first biodistribution study where we fluorescently label the particles, and you can indeed see that they go deep into the lungs in these transgenic mice. And so this is the core team, myself, CTO, CSO, and then Pasi is more like the business finance strategy guy, and then Jani is the medical device expert, and Pei is then like the expert in the microfluidic chip and how to produce them. And this is the next step. We are asking money to go to the first-in-man safety study. And yeah, thank you. Questions?
Thank you very much. Could you maybe give us a first example where your success was so apparent, both for the targeting and also for the delivered subject, and telling us what type of diseases you have in mind as a first indication?
Yeah, so the first indication now we've been making a COVID-like synthetic particle because COVID, as we all know, spread around the whole world. So if you make a COVID-like synthetic particle, you have super high uptake, like less than an hour you get 90% uptake in lung cells, human lung cells. And for that you can have it as a medical device, for the blocking, without API. But then you can use it as a Trojan horse, so to say, and load drugs inside a particle for, let's say, like lung diseases, cystic fibrosis, or antibiotics to the lungs, whereas now you need to eat antibiotics and maybe 1 or 2% goes to the lungs, whereas with our particles you can inhale it and maybe 90% would go to the lungs. So get better effect and less side effects.
Thank you very much. It's very interesting. I'm just wondering, have you done any toxicity on the compounds, or are you planning to do any toxicity tests?
Yeah, so as you can see here, the carrier itself is safe to be administered to human cells, even to 500 micrograms per milliliter. So that's like insane amount. And with the mice study, we also went really high, and there's no issue when you give it to in the nasal repeated dose. So everything looks good. But then, of course, we are now trying to find partners to take your drug that is problematic. Maybe, the IP went bad and you are looking to new, avenues, or your drug is a bit, complicated and has solubility issues. Then we bake it to our particle and we can both party.
We need to move on, and our next speaker is Christian Dweger, who is representing Swiss Immune Amedioo company. And this is a virus-based dual mechanism immuno-oncology idea. And apparently, if I read pre-reads well, there were already several positive examples in the animal application field. So we are really excited to hear about your advances, Christian.
Perfect. Dear Thomas, dear organizers, ladies and gentlemen, dear audience, thank you for inviting and your kind introduction. It's an honor to present actually even some new data today and our approach to take our immuno-oncology technology in animal health, animal immuno-oncology, now into humans in non-core indications outside of head and neck cancers. As you can see from this first slide, and I had to really put it right here in front of you, we have tremendous efficacy as shown on the right-hand side in animal models that we also see in humans. Our technology goes back to more than 20 years of immunological research from leading university hospitals in Europe, primarily in the DACH region. We have a viral vector approach with dual mechanism, coming from a careful selection of antigens and a careful selection of intracellular immunological stimuli, causing cell death only of cancerogenous cells and cancer tissue. As you can see from the skin melanoma, or sarcoids called in horses, we achieved around 74% of complete response in 29 horses, I showed here in February. And actually the centers continued, and we are now standing at 77 horses and reach above 93% of complete response, at above 93% of complete response in horses. We assured financing of 14 million Swiss francs in summer and are fully financed now through to US Department of Agriculture, or FDA, and EMA. We actually now take the same approach from animal health into human health. Setup and key components are identical, as we could not get the CRO support we needed, including AI. From beginning to end, we now partnered with Trialize, a great AI startup company also presenting here in February, and created our own AI-driven Swiss Excel CRO as a purely immuno-oncology specific in-house service provider. Manufacturing is organized, and we do expansion work with an incubator company called Nemo AI Value Builders, now for dogs, cats, camels, and other indications in humans. We're finding value and create ethical financial cases with our ethics-driven concept, validate, invest, and let the companies fly later on, fully focused on their own AGs and GmbHs to their milestones or exits. I come back to the first slide showing the tremendous efficacy both in animals and humans. We see this in animals as well as in humans. Publications exist manifold. Our product needs to be adjusted immunologically to species and cancer types in humans. Our team of molecular bioengineers can do that within short time, actually within a couple of weeks. And we advance the best cases only. A standard approach well done with latest science and AI. As you can see, we achieved everything you see in green already. Phase 1 and Phase 2 transition approval by Paul-Ehrlich Institute just in August 2025 in Germany, hence EMA. For head and neck cancer and cervical cancer. Head and neck cancer is not in our scope. We do not have a right or license for that. For all the rest, we have market comparable levels of license with ethics and compliance ensured. You see in green what we have and where we stand, and actually what we have to do in white boxes to go towards initiation of Phase 3, either as an exit or with a strong partner. You see in green also what we have, and actually you see that we have grouped into 3 groups: pelvic, skin and melanoma, which is huge, and we have all the data and the experience from the animals. Thoracic, and also we group the rare indications in cancer where the big companies normally don't go because you also don't want to have the pricing discussions afterwards if you come with little indications later on. So we are very much about ethics as well. So here, basically, you see the 3 groups. Actually, the investment case is only to take these different 4 groups of cancer types from Phase 1 to Phase 3 and then exit or partner. In bold, you can see where we are between Phase 1 and 2 or even approved by Paul-Ehrlich Institute and EMA already, and where we have strong data to discuss starting directly in phase 2. And basically showing here the risks are covered. And for completeness also, we have all the cards in our hands, a team, a great technology, a great platform. And actually at the end of the day, we also have not only AI, but also websites. So on the weekend or after Gabrielle's presentation after me, you can type that into your mobile phone and it's all in the public domain. It's an execution case, and I thank you for your attention, and I'm open for questions and discussion.
Thank you, Christian. So while we are watching the team slide, maybe I just start with one little question and then we have to move on. And this is your horse results are very, very impressive. Was this intratumoral injection directly, or how do you apply your?
Yeah, actually the technology works systemically as well as intramuscular or in the skin directly. We were playing with at the site of the melanoma or the sarcoid and systemic, and we played a little bit. So I'm now sure that the company here is watching very carefully what I say. And actually, I'm just sharing it for the benefit of horses and humans. We actually think that it's just best to do double or triple injection over time, directly close to the site where you have the tumor. But what we saw in the horses, it's beautiful. You have these Lyme systems. Basically, when you inject, you see the swelling, and the horse has a little bit of elevated temperature, so you see the immunological response. And actually, the virus therapy that we have is eating through the whole horse, actually, into the last corner, we believe, killing these targeted cancer cells.
Our last speaker of the session, so Gabriele Gasch from SAPPIENS SAS. And this is about a neuroprotective treatment in Parkinson.
SAPPIENS, Science Against Parkinson, pharmacological innovation to enhance neuron protection and survival. I'm delighted to introduce CXS003, the neurological flagship program of SAPPIENS. SAPPIENS has been created as a subsidiary of CXS Therapeutics, a platform company dedicated to identify new programs and innovative IP. CXS003 is a patented combination of two anti-diabetics, which is protecting at very specific dosage range. Both molecules taken individually have shown neuroprotective properties and potential, and preclinical results have recently shown the neuroprotective effect of CXS003 by regulating mitochondrial metabolism and decreasing oxidative stress, as well as decreasing neuroinflammation. The suggested mode of action is NRF2 activation, as well as NLRP3 inhibition. To look at some data, I thought it's interesting to show and share some in vitro Parkinson model here. On the left-hand side, you see healthy neurons, then the intoxicated, diseased neurons, where you don't see any activity in red, and then the diseased neurons. And if you add CXS003, you see that there is kind of restoration of the dopaminergic activity and production. This interesting data has been confirmed in vivo models, where we have taken very diseased, intoxicated cells to show very advanced, to mimic advanced Parkinson's disease. The major effect here is significant results to show that the combination of both molecules has a significant effect, whereas each molecule alone has no effect at all. In another model, here in Parkinson model, you see that the neuron survival in the brain is significantly superior with CX-003. The team around the development program has multiple years of experience in the industry in clinical development, is surrounded by scientific experts for preclinical development, and clinical experts will be joining as we go forward in the adventure. We are supported by scientific partners as well from academics as industry. We are currently raising 3 million euros to finalize the preclinical package and have a solid translational program to go before entering into phase 1/2. And we believe that CX-003 as add-on therapy to current standard of care will have significant potential and important return on investment. Happy to take your questions later during the course. Thank you.
Thank you very much. Maybe I can start with one question and then the audience is coming. And this is, I was a bit surprised by the choice of your medications. So I would like to ask you whether you had some epidemiological evidence that there is a kind of relationship between diabetes and Parkinson's, and the consequence of that is there a change in glucose metabolism in Parkinson's, oxidative phosphorylation, for example, which can be directly then addressed by these drugs?
So the choice of the two molecules has been done by very thorough review of bioinformatic platform and literature review. And the combination of both in various modeling has given this indication to combine metformin with glibenclamide. The various and the very important reasoning behind is the ratio of both molecules in combination, and this is really critical for the clinical development. We are talking about very low dose combination therapy here, which shows this effect in human. To answer the second part of the question, we don't have all the proofs now, but we are working on it.
I was just wondering if you also had data from models where you use more, aggregation-dependent models, like alpha-synuclein de novo aggregation or PFF induction, or if it only relies on toxin models such as 6-OHDA and rotenone and those kind of.
For the time being, these are the models we have chosen.
So, thank you. So first of all, I wanted to thank again to the speakers who did a great job also keeping the time, which is fantastic. Audience, thank you very much for keeping also us in your attention and for some of the questions. And last but not least, thank you also the organizers who made this session possible. And finally, we are concluding a great conference. So all the best and see you next time.